Okay, so today's second practical is bioassay of serotonin. So let's see. The purpose of today's experiment is at the end of practical class, the student shall be able to know the detailed procedure of 3-point bioassay. Second purpose is to know about the advantages and disadvantages of 3-point bioassay. Third one is to know the pharmacology of serotonin, and the fourth one is to know the procedure of rat fundus strip preparation. Okay, so from the objectives only we can get few things. That is, the first, the method which we will be using is 3-point bioassay. Then second thing is that we are supposed to go into details of the pharmacology of serotonin. So you should be able to understand the serotonin, where it acts and when it binds to particular receptors, what effect it produces. That all you should be able to recall. And the next is that rat fundus strip preparation. So you have to first of all locate where the fundus is present, and once you locate it you have to isolate the fundus and you have to prepare the strip of it. Okay, so not too much difficult is this to prepare this fundus strip preparation, but you have to first of all identify which organ or which system consists of this part, that is fundus. Okay, so terminology, that is the first one is the 3-point bioassay. So it is a type of graded bioassay in which two responses of standard drug and one response of test sample, they are selected such that the test response is intermediate between the two responses of standard drug and all responses are in the linear range of the DRC. Okay, so what we mean by this, that the height of one standard response should be less than that of the height of the test, and of the other standard response the height should be greater than that of the test. So the test height, it should lie between the two standards. So this will be the S1, this will be the S2, and this will be the T. So the height of the T, it should lie between the height of S1 and height of S2. So this S1 and S2, they are like the extremities of a range. Okay, and in between these two extremities the value of T lies. Okay, so the response, they are repeated several times and the mean of each is taken. Okay, so the concentration of unknown test sample is determined graphically as well as mathematically. So description. So 5-hydroxytryptamine, that is 5-HT, is the biologically active local hormone. It has lower molecular weight and it is also important neurotransmitter in the brain as well as in the periphery. It was first discovered or detected in serum, that's why the name serotonin, and in the enterochromaffin cells of the gut mucosa, and that's why it is also called as enteramine. So later both were identified to be 5-HT. So earlier on it was thought that the serotonin is different from that of the enteramine, but when chemical analysis was done, it was found that it is the same substance and it is named as the 5-HT. So today the terms 5-HT and serotonin, they are used interchangeably. Okay, so about 90% of the body's content of 5-HT, it is localized in the intestine and most of the rest is present within the platelets and in the brain. So it is also found in the wasp and the scorpion sting. and is widely distributed in invertebrates and the plants. So in plants you can find it in banana, then pear, then pineapple, tomato, etc. Okay. So 5-HT, it is formed from the dietary tryptophan, which is converted to the 5-hydroxytryptophan by the enzyme, that is tryptophan hydroxylase. So this is biosynthesis of the 5-HT that we are talking about. And then it is this 5-hydroxytryptophan converted to the 5-hydroxytryptamine by a non-specific enzyme, that is decarboxylase enzyme. So 5-HT, it is transported into cell by a specific transport system. So degradation, it occurs mainly by the enzyme, that is monoamine oxidase, and specific isoform of it, that is monoamine oxidase A. And when it is acted upon by the monoamine oxidase, it forms 5-hydroxy indole acetic acid, that is 5-HIAA, which is excreted in urine. Okay, so important actions of 5-HT, they are increased gastrointestinal motility, then contraction of the other smooth muscles, that is bronchi and uterus, then mixture of vascular constriction, where direct and via sympathetic innervation, and then dilatation, that which is endothelium dependent, then platelet aggregation, which takes place in case of tissue injury, then stimulation of the peripheral nociceptive nerve endings, and excitation and inhibition of central nervous system neurons. Okay, so next, the 5-HT receptor, they are of 7 types, that is 5-HT1 to 5-HT7, and which further subtypes we can find of the 5-HT1, that is from A to F, except for C, and the 5-HT2 we have 3 subtypes, that is A, B and C. Okay, so all of these receptors, they are G protein coupled receptor, but there is an exception to this, that is of the 5-HT3, which is a LGIC, that is ligand gated cation channel. So physiological and pathophysiological roles of the 5-HT in periphery, it includes peristalsis, vomiting, platelet aggregation, hemostasis, inflammatory mediator, sensitization of the nociceptors, and microvascular control. And in CNS, many postulated functions, they include control of appetite, sleep, mood, hallucinations, stereotype behavior, pain perception, and the vomiting. Okay, so most of these functions, as we can see, they are very important, because whatever drugs that are acting on this serotonergic system, they either potentiate these effects or they inhibit these particular actions. Like the specific drug that we have studied, that is a ondansetron, it inhibits the vomiting, okay, by acting on to the specific 5-HT receptors. So the clinical conditions which are associated with disturbed 5-HT function, they include migraine, then carcinoid syndrome, which is a tumor of the cells of the gastric mucosa, then mood disorders, and the anxiety. So in all these conditions, it was found that there is some abnormality of the serotonergic system. Okay, so concentration of 5-HT in biological samples, it can be assayed by various bioassay methods. They include, the first one is isolated fundus strip of rat stomach, then second is isolated terminal colon of rat, then isolated rat uterus, and lastly the perfused rabbit ear. Okay, so the first 3 methods, they are in vitro methods, and the last one is the in vivo. Okay, So objective is to determine the concentration of serotonin by 3-point bioassay method using rat fundus strip preparation. So principle, rat fundus strip is a very sensitive tissue for the bioassay methods, and it is useful to study action of various substances like 5-HT, SEH, then PGE2, that is prostaglandin E2, and lastly the bradykinin. Okay, so we have already studied about the pharmacology of the 5-HT and SEH, and next chapter we will be studying about PGE2, and then we will be studying about bradykinin. Okay, so fundus strip preparation is slow contracting muscles, so that's why the when we will be talking about time cycle, the time which is allowed for the drug to remain in contact with the tissue will be more, that is contact time will be more. Okay, so fundus part of the stomach, it can be easily identified by its gray color, and it is situated above the pink colored thick pyloric part. Okay, a zigzag preparation of the fundal strip is prepared so as to expose maximum portion of the tissue to the drugs, which is shown in this figure. So you can see this is the rat stomach, then fundus of the stomach is this upper portion. You can see it is like gray colored portion. We have to cut the fundus from the lesser curvature, and so that it will get open, and once it is open we have to alternatively make the cuts into it. Okay, so that it will look something like this, and this is nothing but the fundus strip preparation. Okay, so cut should be incomplete. Okay, please remember, cuts that we will make into the fundus strip, they should be incomplete. If we cut it completely, then we will get separate pieces of the fundus strip, and which are useless. Okay, so alternatively we have to cut, and we have to cut it incompletely. Okay, so the tissue is sensitive to 1 nanogram per ml of the serotonin and 0.2 to 0.5 nanogram per ml of the acetylcholine, and that's why it is said that it is very sensitive tissue preparation. Okay, so in 3-point or 2+1 bioassay, 2 doses of the standard and 1 dose of the test, they are used. So the test response should be intermediate between 2 responses of the standard drug. Okay, so all the 3 responses should be in the linear range of the DRC. So the responses, they are repeated several times with a sequence in a random fashion of Latin square design, and mean of each is then taken. So the concentration of unknown test sample is determined graphically, or it is determined mathematically. So requirements are, the animal, we have to take a rat, weight should be 180 to 200 grams, and the tissue that we have to take, it is rat fundus strip muscle. Then drugs, we have to use standard solution of serotonin. The molecular weight of it is 176.22, and test solution of serotonin, the concentration is unknown. The PSS that we have to use, that is Krebs solution. Then equipment that we have to use, chymograph with drum, then smoke paper. In our case, we don't have to go for smoke paper. Instead of it, we can take the white paper. Then student organ bath, isotonic frontal writing lever, then aerator with aeration tube, and other accessories. So other things that we have to take, that is tuberculin syringe, the beaker, stopwatch, petri dish, dissection box, and suturing needle and the thread. So procedure, so first of all we are going to prepare the tissue, and then we will actually mount it into the organ tube, and then we will actually perform the experiment and do the calculations. Okay, so we have to prepare the stock of Krebs, then we have to prepare the stock solution of serotonin whose concentration is 1 milligram per ml, and different concentration of serotonin we have to make by the serial dilution method. Okay, so we have to sacrifice 24-hour fasted rat by stunning on the head and carotid bleeding. Fix the animal on the dissecting board by tying its legs. Open the abdominal cavity by small horizontal cut followed by vertical midline incision. Expose the abdominal organs. Then we have to identify the stomach. We have to separate it from the abdomen by gently cutting its cardiac and pyloric end. Then we have to place it in the petri dish which contains aerated warm grape solution. Then we have to incise the fundus of the stomach, which is nothing but the upper gray part from the pyloric part, which is pink and thick part. We have to cut the fundus from lesser curvature and open it longitudinally. We have to cut the fundus at the midline into two equal parts. Give alternate transverse cuts, that is zigzag cut, on opposite sides of the muscle to make the fundal strip preparation. Okay, so we have to mount the preparation in inner organ bath which already contains grape solution, which is around 20 ml, which is maintained at 37 degrees centigrade. We have to tie the bottom end of the muscle to the hook of aeration tube and upper end to the isotonic frontal lever by a thread. We have to adjust the magnification response to 10 to 15 fold. These values are very important, and we have to aerate the tissue with the O2 or the carbogen slowly, which is 40 to 60 bubbles per minute. So we have to apply 1 gram load. Remember, in earlier experiment we used 0.5 gram. Here we are using 1 gram. So depending on the type of tissue, the load also varies. Okay, so we have to stabilize the tissue for 30 minutes, during which we have to wash the tissue with fresh grape solution once in every 10 minutes. So in total we have to give 3 washings. Okay, so we have to use 5-minute time cycle with a contact time of 90 seconds. As I said earlier, as the tissue is slow contracting, we have to allow the maximum time for drug to remain in contact with the tissue. Okay, so recording the contraction due to serotonin on the smoke drum as per the following schedule. So at 0 seconds we have to remove the weight, start the drum, and we have to record the baseline. So at 30 seconds we have to add serotonin in inner bath and record the response for 90 seconds, that is the contact time. So at 120 seconds we have to stop the drum, we have to add the weight, and we have to wash the inner tube. So at 210 seconds we have to give the second wash, and at 300 seconds we have to give the third wash, and we have to remove the weight, and we have to start the drum. Okay, so we have to record the graded response of the standard solution of serotonin in geometric progression on the fundal strip preparation till getting the maximal effect. So that is DRC of standard we have to complete first. Then we have to select 2 doses of the standard solution as S1 and S2, that is 1:2 dose ratio, producing corresponding responses, that is capital S1 and capital S2, which should be in linear range of the DRC. Okay, so understand, dose we are denoting as small s1 and small s2, and the responses we are denoting as capital S1 and capital S2. Okay, so we have to record the response due to unknown concentration of test solution with gradually increasing the volume. We have to select the test dose, that is T, producing the response capital T, which should be intermediate between the responses, that is capital S1 and capital S2. So the sequences of all 3 responses should be S1 should be less than capital T, and T should be less than capital S2. Okay, so we have to standardize the tissue with the dose of the S1. The tissue is said to be standardized if it responds equally to the same concentration. Okay, so earlier on when we had taken the S1 dose response, whatever height it produced, that is the capital S1, after during standardization process it should produce the same amount of height, or to same extent it should produce the response. Then we can say that the tissue is standardized, and then only we can use the tissue for the further experiment. Okay, so we have to record 3 sets of responses due to above selected dose, that is S1, S2 and T, on the chymograph drum with a sequence as per the Latin square method of randomization, which is shown below. So in this fashion we have to give the response. So first is T, then small s1, then s2, then we have to move s1, s2, small t, then s2, t and s1. Okay, so in total how many responses? 1 2 3. So total 9 responses we have to take as per Latin square design. Okay, so procedure we continue, that is we have to label and fix the dose response graph. We have to measure the height of all the responses, and we have to calculate the mean of each response, that is of S1, S2 and capital T. So the mean response is plotted against log dose, and the amount of standard producing the same response as produced by the test is determined graphically as well as mathematically by using the formula, which is relative potency, that is M is equal to capital T minus S1 divided by S2 minus S1 into log S2 divided by S1. So potency ratio we can get is equal to S2 divided by T into antilog of capital M. So we have to first of all determine this capital M, and then we can get this value, that is potency ratio. Okay, so CT is equal to potency ratio into CS. CT stands for the concentration of test, and as we already know the concentration of standard, we can just multiply it with that of the potency ratio. Okay, so these are the graphs that we will get. So first of all we have to take a DRC of standard, then of test, and then 9 responses of the two standard doses and 1 test response. So 1 2 3 4 5 6 7 8 9. So in total 9 responses we have to take, and graph like this we have to plot, where the percent of the response is on the y-axis, and on the x-axis we have to plot the log dose points. Okay, so this is the observation table that we have to maintain. So different responses of S1, S2 and T. So 3 responses of each S1, S2 and T, and we have to take mean of it. Okay, and we have to use appropriate mathematical formula and then calculate the relative potency and concentration of test sample. So inference that we will take out from this experiment is that the 3-point bioassay is more reliable method, and the chances of error in this bioassay is less as compared to the matching bioassay. Why? Because the sensitivity of the tissue is first determined by plotting the various responses with the known agonist. So the 3-point bioassay, where 2 responses from standard drug and 1 response from the test sample, they are repeated several times with a sequence in a random fashion of Latin square design. The mean response of each is calculated, and the concentration of unknown test sample may be determined by graphical or mathematical method. Okay, also the potency ratio of the test compound, it may be calculated. Okay, so these are the questions. So you have to write down these questions in your notebook, and you have to solve these questions, and you have to submit it into the clever. I will give you like the 30 seconds to write these questions, and then we will stop with today's experiment. Okay, I hope that you got all the questions in your notebook. Solve it and upload it into the clever. So that was all from my side for today's experiment, and we should stop over here. Thank you.